Hello, everyone. My name is Manolis-Kellis, and today I'd like to tell you our work translating genomics into therapeutics by dissection and manipulation of human disease circuitry at single-cell resolution. Our work starts with disease genetics. We start with common and rare variants that are associated across the genome with various disorders. We then profile RNA and epigenomics in both healthy and diseased individuals across thousands of samples to understand the differences that are associated with genetic differences between individuals, but that are also associated with disease conditions. We then integrate the data computationally to predict using causal models and deep learning models and Bayesian models the driver genes, regions, and cell types that are responsible for the phenotypic variation using the intermediate molecular phenotypes as evidence to understand the pathways and the biological processes that are involved. Having done that, we validate our predictions in human cells and in mouse models through a number of collaborations for every distinct disorder that our lab works on. We disseminate the results and we start the cycle all over again. My own lab has generated one of the largest collections of human single-cell data sets in the context of disease. We've done that for a number of brain disorders, including Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in AD, schizophrenia, bipolar disorder, Down syndrome, autism, depression, suicide, and PTSD. But we've also looked at many non-brain disorders, including obesity and exercise, cardiovascular, cancer, and overall aging. We've done that for many different traits, each across dozens of individuals for a total of more than 1,500 samples. We've looked at many different cell types, more than 60 different cell types in the human brain, and more than 100 different cell types across multiple different organs, and also looking across many individual cells, more than 20 million cells have been profiled in my lab with single cell maps, both RNA and epigenomic. And we've done that across many different brain regions, across heart, muscle, adipose, etc. We then developed a large number of computational methods for integrating the data. We basically developed Bayesian methods and causal inference methods, convolutional neural networks, and other deep learning representations for inferring the language of DNA and also being able to translate between RNA, epigenomic, and genetic information, and methods for causality inference and mediation analysis. These methods have revealed circuits for 30,000 disease loci, and these circuits tell us about the upstream regulators, the causal variants, the downstream target genes, and ultimately the pathways and the cell types where these genetic variants associated with disease are acting. Why are we excited about circuits? Because when we know the circuitry, we can manipulate the circuitry to reverse the disease phenotypes. In the case of obesity, we uncovered the strongest genetic signal underlying obesity and its unique circuitry, which allowed us to now start intervening and manipulating this circuitry to switch back and forth between lean and obese phenotypes. We've done that in the case of CRISPR editing of human primary adipocytes from obesity risk individuals to restore the process of thermogenesis that we discovered underlies this strong effect locus. And we were able to then go and knock down the downstream target gene that was overexpressed in obese individuals to show that the mice, in fact, lose 50% of their body weight and completely deplete all their fat stores by shutting off the circuit of lipogenesis and instead turning off the circuit of thermogenesis, whereby all of the excess calories in the diet of these mice is in fact burned off as heat. And you can see that in a high-fat diet, these mice are unable to gain weight. This is what we would like to do systematically across thousands of disease loci. To do this, our lab has scaled up across hundreds of epigenomic datasets, from the Roadmap Epigenomics reference that we published in 2015 to the EpiMap reference that just appeared in Nature just a few weeks ago. We've now gone from 54 enriched GWAS traits to 534 different traits, giving rise to 30,000 individual SNPs, individual single nucleotide variants, and disease loci that are now dissected based on their unique location and circuitry. This allows us to understand the tissues and cell types that are responsible for many different disorders. For example, looking at depression, neuroticism, schizophrenia, as well as, of course, intelligence, self-reported motility that localize in the brain, looking at Alzheimer's that surprisingly localizes in the immune cells, giving us rise to this immune hypothesis for Alzheimer's that our lab has worked very strongly on, looking at liver and cholesterol, and looking at diverse cardiac traits. I want to point your attention in particular to the fact that many of these traits are don't show just one color, don't show just one tissue of association, but appear to be involved in many different tissues, and in particular coronary artery disease. This was in fact by far the most pleiotropic trait, giving rise to enrichments in 19 different diverse tissue groups across our circuitry. That allows us to now start partitioning the complexity of coronary artery disease into each of the tissues that are enriched and taking the subset of genetic variants that overlap liver, coronary artery, thyroid, adipose, and so on and so forth. That allows us to now start understanding the distinct pathways associated with genetic variants that overlap distinct epigenomic activity patterns. What does that mean? That means that we can take very complex traits and break them up, partition them into their simpler parts. What we can also do is go into individual genetic loci associated with these traits and start asking across the enriched annotations, where does this trait, where does this locus appear to be acting on this trait? We find examples such as PCSK9, which appears to be acting in liver, and this is a well-known mechanism that has now led to a new therapeutic. EDNRA, which appears to be acting specifically in the heart, as well as PLPP3, which appears to be acting in both liver and coronary artery disease. And we've now constructed these annotations for 30,000 disease loci that you can go and browse freely at compile.mit.edu/epimap. So having that ability to now look at each of these loci and their circuitry gives us a handle for building these therapeutics across thousands of loci. We've also developed many methods and data sets for single cell dissection of disease, looking at both non-AD, mild AD, and severe Alzheimer's individuals across the progression of disease, allows us to now start asking, how does every single cell, which is a dot in this picture, change with respect to the sex of the individual, the age, many different pathological variables, and so on and so forth. One of the big surprises was that female individuals appear to be much more associated with disease data sets, and male versus female individuals show thousands of differentially expressed genes, including myelination pathways that appear to be increased in response to AD in male individuals, but not in female individuals. And indeed, we do find that the female individuals show an increase in myelination loss and white matter loss in their pathology. We've expanded these studies to many different disorders. For example, in schizophrenia, we are able to distinguish many different subtypes of excitatory and inhibitory neurons in the cortex that show dramatic transcriptional alterations in cases versus controls. And many of these actually land and help explain genetic loci that are associated with schizophrenia, enabling us to predict both the cell type of action, the target gene, and the directionality of effect, which allows us to now therapeutically know which genes we should be overexpressing and which genes we should be repressing therapeutically. We can also find that there are in fact two different paths to schizophrenia. One is a global path of dysregulation across all of these major cell types. Another one does not show this global dysregulation, but instead shows an abundance of only one cellular state that appears to be significantly enriched in schizophrenia individuals. We're able to start understanding the upstream regulators that control many of these processes. And what we find is that they act both early in development and in adulthood, suggesting pleiotropic functions of many of these loci, enabling us to now start understanding how an early developmental disorder can also have a late life manifestation. We've also started looking at how the brains of these individuals change across different brain regions. And we're able to now start painting a map of many different subtypes of excitatory and inhibitory neurons, including 30 different subtypes of excitatory neurons and 23 different subtypes of inhibitory neurons across these major regions of the brain. We're finding that both neuronal cells and glial cells vary dramatically across cell types. And we're also able to study the progression of Alzheimer's disease across these individuals, across these regions, and across individual cells, enabling us to discover genes that act early in the disease, intermediate, and late in the disease process, and the specific cell type in which they are acting. Computationally, we've been also able to show that we can capture spatial transcriptomic information at single cell resolution by being able to computationally subcluster regions of the hippocampus and regions of the entorhinal cortex to regions that are very strongly affected in Alzheimer's disease using their single cell profiles, even though we did not require to have spatial information directly. We've also been able to couple our single cell data with spatial information, what I like to call mini bulk profiles, to deconvolve each of these mini bulk experiments into the specific cell types that are acting, showing that indeed there's a large loss of Purkinje cells in SCA1 in the in specific regions of the brain, in the cerebellum in this particular case. We've also been able to augment single cell data sets across millions of cells using combined spatial transcriptomic and single cell profiles by training a deep learning model that allows us to learn from the single cell data what are the features that are responsible for predicting the position of each of these cells across thousands of cells, and then expanding that model and applying it systematically across millions of cells. Focusing in on specific cell types, we're able to find specific subtypes of inflammatory or synaptic microglia that are very differentially enriched in Alzheimer's disease versus schizophrenia. They play roles in both, but each subtype appears to be uniquely acting in one or the other. And we've also studied the vasculature and the blood-brain barrier at single cell resolution across tens of thousands of cells, enabling us to study the patterning and the zonation that happens between the arterial and the venial for multiple cell types, including endothelial cells, smooth muscle cells, and pericytes. We've also been studying not just the changes in RNA, but also changes in DNA accessibility to look at concomitant changes that are happening between disease and healthy individuals, affecting multiple cell types and affecting both transcriptional and epigenomic signatures. And we're also able to use the single cell DNA accessibility information to recognize the cell types responsible for diverse psychiatric, neurodegenerative, and also other phenotypes acting in brain and in diverse cell types, including recognizing regions that are uniquely altered in very specific cell types and the regulators that are acting upstream of each of these regions. Going beyond just the brain and beyond individual tissues, we're also looking at the multi-tissue, multi-omic changes that are happening in response to exercise and diet in both human and mouse. We're profiling the brain, as well as the intestine, the visceral and subcutaneous fat, muscle, liver, heart, and blood across both humans and mice in response to exercise and diet interventions, discovering 35 major cell types across hundreds of thousands of cells, and how these change across tissues and how these change across phenotypes. We're finding that dramatic changes are happening in both T cells and in adipose stem cells, affecting both immune processes and affecting the communication channels between pairs of cells, enabling us to actually study the inverse effect of training versus exercise, whereby obesity induces a large number of immune interactions with other cells, whereas training, exercise, in fact, appears to be suppressing those. And we're also able to look at longitudinal changes that are happening in patients at single cell resolution, in this particular case, in the context of cancer. We've also published on the analysis of electronic health records and medical records in the context of their genetic information. And we've also developed high throughput experimental methods for testing the millions of predictions that we're making in experiments that allow us to carry out 7 million tests in a single experiment. And in several collaborations, we're now building these modular and programmable CRISPR-Cas9 based intervention systems, enabling us to modulate each of these processes in specific cell types, specific targets, specific induction, specific environmental contributors, and looking at multi-cell type effects. So overall, I would like to point out that many of our students have now taken on their own faculty jobs. We've published extensively across some very high impact journals. We have hundreds of citations every month and many active grants. So overall, we are eager to work with you guys. Come join us. We are at the interface of big data, generating much of that ourselves, machine learning integration, experimental validation, leading to a lot of impact and a lot of discovery. So I'm only one of many faculty members at MIT who are working at the interface between genomics, medical information, neuroscience, proteomics, synthetic biology, and much, much more. And MIT has really become a mecca and a nexus for biotech, pharmaceutical, and innovation overall. And I encourage you to join our panel on AI, perturbation, integration, and discovery, where several of our faculty and also several members of the biotech community and the pharma community in Boston will be speaking this Thursday at 3:30 to 5 p.m. Thank you very much.